Okay, fantastic. Well, thanks everyone for getting on. For this is the Judy Cho as the second speaker for the Institute for Precision Medicine's Fall 2023 Speaker Series. As you all know, one of our goals is to seek out and hear from other institutes for personalized or precision medicine, and Judy certainly fits this role. Judy began her education at Ohio State University, where she received her bachelor's, followed by her MD at OSU College of Medicine. She then went on to do postdoctoral training at Northwestern University and the University of Chicago. Currently, Judy is the director of the Charles Bronfman Institute for Personalized Medicine, the Ward Coleman Chair for Translational Genetics, and the Vice Chair for Research in Gastroenterology at the Icahn School of Medicine at Mount Sinai. Over the past 20 years, Judy has been a principal investigator on over 100 funded projects, is an author on over 700 publications, and holds 3 patents. Notably, she was one of the leaders of the team that identified the first gene implicated in Crohn's disease, NOD2, followed by identifying the involvement of IL-23 in the disease cascade. Judy has an impressive list of honors and awards that would be impossible to do before she gives her talk. It would take over all the time. But those awards demonstrate her dedication to promote research finding to clinical care advances. In 2021, she received the prestigious Sherman Prize that recognizes recipients who represent excellence in Crohn's and colitis in their chosen endeavors, having dedicated their careers to the fight to overcome IBD. Judy's research focuses on a wide range of themes related to the genetics, environmental factors associated with IBD, which she will talk about today in her talk entitled Precision Inflammatory Bowel Disease, IBD, and Research Within Health Systems, Accelerating Research to Clinical Translation. Judy, thanks for speaking today.
Thanks so much, Adrian, for the kind invitation. So it's a real pleasure to talk on precision medicine. And so what I'll cover in today's talk are 3 topics. One is just general comments about the value of biobanks. Second is most of what my research focuses on, which is genetics and single-cell analysis of inflammatory bowel disease, or IBD. And I'll close with just a few brief comments on the value of diversity in precision medicine. So part 1, biobanks. And so there are a variety of biobanks of which these are listed probably in decreasing significance in terms of their prominence in the research realm. The UK Biobank has been truly transformative. It's provided enormous replication capacity, both within the UK and throughout the world. The All of Us Biobank is rapidly catching up. There are a large number of investigators throughout the country that use this. It has the advantages of diversity and the use of not just health systems, but also direct-to-patient use of biobanks. The third major biobank is the Million Veterans Program, which is a wonderful biobank. It also has the advantage of an integrated electronic health system. There are many other country-based biobanks and many health system and medical school biobanks that feed into either a standalone or feed into these other ones. And to a large extent, most of the research that's come from these biobanks have been DNA-based in genetics. Although with a lot of the emerging platforms, I anticipate that some of the plasma-based biomarkers will maybe of value in the future. So in terms of the Mount Sinai BioMe Biobank, the BioMe Biobank is an electronic health record, or EHR-linked biobank, that has the capacity to mine the electronic health record, both retrospectively and prospectively, with questionnaire data. So I actually stepped down as the Charles Bronfman Institute for Personalized Medicine director about a year ago. And right now, BioMe, the Mount Sinai Biobank, has about 65,000 participants with DNA and plasma. And the advantages of the Mount Sinai Biobank is the advantages of doing research in New York City. Here's a picture of Manhattan. And the BioMe Biobank has traditionally been about 40% Latinx, 35% European and gray, 25% African American. And you can see as you look at the north-south distribution throughout Manhattan, the distribution of the demographics throughout the island that I live in. And so they're consented for the mining of the EHR, and overwhelmingly, one of the big advantages that we've actually leveraged quite effectively in BioMe is the permission to recontact, and this has benefited a number of different faculty throughout Mount Sinai, so that if someone finds a mutation of interest and they want to recall and do additional studies with those patients, the Mount Sinai BioMe consent allows that to happen. And again, I'm happy to take any questions during this. I just saw a chat thing. Yes. Okay. So happy to take any questions, and if someone could just break and tell me that there's a question in case I missed that. So the BioMe Biobank, as well as any other biobank within the U.S., I think has similar strengths and weaknesses. Especially in New York City, it reflects the patterns of healthcare delivery, which is often quite fragmented in nature. It's very normal in New York City for some patients to have their care of one particular issue at one location and another one. So it's an often fragmented structure of healthcare that may not necessarily get the whole picture of the patient. What has been achieved very successfully in a number of papers coming out of BioMe is use of leveraging of structured data, and we've begun to do some text mining. In addition, Mount Sinai has successfully instituted the OMOP model, which is a structured database structure that allows you to more effectively mine the electronic health records. In my opinion, point number 4 in terms of medication use, it's very hard to mine. It really is one of the biggest questions that one might want to answer is to look at therapeutic response, and that's traditionally something that I don't think you can actually effectively do easily within these large health system-based biobanks. And finally, the family history is inconsistently recorded and acted upon. What is easy and what a number of investigators at Mount Sinai are leveraging very effectively are imaging. In radiology, increasingly healthcare is increasingly non-invasive in its nature, and your capacity to sample a rich amount of imaging, I think, is a treasure trove of potential future opportunities. And so radiology, you can think of this as being the mining of just the reports, which in and of itself, I think, is incredibly valuable. So you can basically take the reports and mine the SNOMED terms, and as well as also do primary image analysis. And so again, what the radiology group has done here successfully is be able to peel off the PHI information attached to the radiology images. So that's been a very effective use of the biobanks and the various clinical resources that are available in the Mount Sinai Health System. So radiology is happening now quite actively, and I think what's going to be an enormous growth area is in pathology. And so at Mount Sinai, in clinical care of pathology, right now, all of the samples are being digitized, which provides an enormous treasure trove of information. And then beyond going past just the H&E stains of what's normally captured within the course of normal clinical care, the other resource that is available to Mount Sinai, the research community, are the capacity to go back. Medical legally, we have to keep the paraffin block resources. And Mount Sinai has allowed investigators to then go back to these paraffin block resources, and you can basically develop scrolls for additional research analysis. Obviously, there are very strict rules about maintenance as the minimal requirement for clinical memorialization. But this is also another very valuable form of research that can be attached to the genetics. And so when you could, another advantage of this is looking forward. This is, again, doing research within a health system is it allows you to do longitudinal research at a relatively low cost. And so an important consideration is looking at trajectories, prediction, and machine learning. And so in the cancer field, obviously, the major goal is to prevent disease. There are certain standard clinical paradigms that are used. And then also, what's remarkable to me is that to a large extent, the standard blood test that's attained in a primary care practice is pretty much the same as it was when I went to medical school over 30 years ago. And so I do think that there's enormous opportunities for research and developing new laboratory-based tests and biomarkers for disease prediction and treatment responses. And so in terms of the cancer paradigm, we're in the process of trying to integrate more with the Cancer Center here at Mount Sinai. This is an older and a complex graph, but it's not just the genetics. You can also look at family history. And so we're plotting here on the x-axis, the risk for cancer, breast and colon. And the red curves designate when you should start looking at high risk. So the polygenic risk scores obviously are going to be most valuable kind of in the younger age ranges. But you can see that both family history and polygenic risk scores provide additive value in terms of providing better predictive capacity for patients within a health system. So we'll start switching to inflammatory bowel disease, which many people would consider to be an autoimmune disease. And so when you think about the time course of disease, starting from the germline genetic architecture, which is the basis for a lot of the biobank-based data, in the case of the various different organs that are affected by these chronic inflammatory diseases, the red arrows designate when a patient presents with disease. But prior to that disease presentation, there's a lot of things that are happening to the left in terms of epigenetics and early exposures, altered immune function, development of autoantibodies. And altogether, in the case of traits like type 1 diabetes and autoimmune thyroiditis, this often happens asymptomatically. And so you don't actually present until the cumulative structural damage has resulted in complete damage to the gland. Other diseases like inflammatory bowel disease, lupus, and rheumatoid arthritis, you have these very active disease flares. And so one of the goals of medical therapy is to prevent the cumulative structural damage that results in the need for surgeries. And so, again, what's similar to the cancer paradigm, I think what's happening in autoimmune disease is that there is a desire to achieve complete control of inflammatory response early on in the disease to prevent this development of cumulative structural damage. So I'm going to shift now to talk about IBD and our 2-cell modules. So inflammatory bowel disease. So a module can be defined in the course of independent units that together can construct a more complex structure. It can be applied to spacecraft. It can be applied to computing. And what I think about a lot is these biologic modules of inflammatory bowel disease. And so the high level classifications of cells that make up the gut are comprised of epithelial cells, immune cells, and stromal cells. And so a very important biologic concept that our lab is really very interested in is looking at these heterogeneous cell type interactions. So just a little bit in terms of kind of the clinical features of inflammatory bowel disease. So it affects anywhere from 1 out of 100 to 1 out of 200 individuals within the United States. It comes in 2 major varieties, Crohn's disease and ulcerative colitis, CD and UC, that are distinguished by different distributions of the inflammation. The peak age of onset, for reasons that are incompletely understood, are between the ages of 15 and 30. There's substantial genetic overlap between CD and UC. You have many families that have one member with Crohn's disease and another member with ulcerative colitis. In many cases, this distinction is driven by tobacco differences, environmental factors, where the smoker will have Crohn's disease and the non-smoker or the ex-smoker with ulcerative colitis. Genome-wide association studies, or GWAS, have identified over 400 IBD loci. The prevalence is highest in Europeans, and it's very interesting that we have markedly different genetic architectures between European ancestry and East Asian individuals. And increasingly, we are practicing precision IBD. The treatment paths are increasingly diverging, and anti-TNF agents are disease-modifying and beneficial, especially in Crohn's disease. Why is this not going here? Okay, oops, didn't go there. All right, so we'll talk about 3 different 2-cell modules. The first is myeloid cells and stromal cell crosstalk and altered blood monocyte differentiation. And so in collaboration with Gabriel Nunez over 20 years ago, we and other genetics groups identified associations of these relatively uncommon variants. So these risk alleles were in the range of 2 to 4% in controls, with markedly increased risk in patients with Crohn's disease. The major mutations were all clustered within the pink peptidoglycan sensing domain of NOD2, which resulted in an impaired capacity to appropriately respond to muramyl dipeptide, which is the minimal bioactive component, or MDP, of bacterial peptidoglycan. And so we think that NOD2 is expressed in multiple cells, but we think that a lot of its major pathogenic effects are mediated via myeloid cells. So we published this in collaboration with the Miriam Murad's lab a few years ago. where we then wanted to look at, this was one of our first forays into the use of single cell analysis. And so we performed single cell analysis of inflamed to the left in red and uninflamed to the right in blue tissues of patients at their resection of the terminal ileum, the last part of the small intestine, at the time of resectional surgery for complications, typically either obstruction or fistula. And so Effie Koenigsberg led the analysis of the single cell. And it was an unbiased analysis. We did not attempt to enrich for any particular cell types. And so at the time, we identified 47 clusters that are colored by these immune cell subsets. And you can see, unsurprisingly, that, again, this is unenriched. We just basically took all comers and sequenced them. And for the adaptive immune cells, the T cells, B cells, and plasma cells were by far the most abundant. But I want to direct your attention to these mononuclear phagocytes, or the myeloid cells, and the stromal cells. And one of the first kind of lessons of single cell is that some of these uncommon cells that are relatively non-abundant compared to the adaptive immune cells can be very highly impactful. And so in the upper right-hand corner, we're showing a principal components analysis. And what was striking is that in equally inflamed regions between patients, we were able to, they really seemed to cluster into two different subsets, a GIMATS high cohort and a GIMATS low, GIMATS standing for IgG producing plasmablasts, inflammatory mononuclear cells, and activated T and stromal cells. And so what drove this is looking at the frequency of those 47 clusters that drives it between the two groups. Was this composite score. And you can see that the x-axis on that GIMATS score heat map is comprised of 84 genes. And a large majority of what's driving this heterogeneity are these green cells, the mononuclear phagocytes, and the stromal cells in brown. In addition, another characteristic feature is that the GIMATS high cohort. Now, the other point to be made is that the individuals with a high GIMATS score, which we then projected onto a bulk RNA-seq data set where we had responses to this anti-TNF therapy, which is central to Crohn's disease. The GIMATS high individuals kind of correlated and went together with folks who were more likely to not respond to anti-TNF therapy. And the final point to be made here is that when we looked at the peripheral blood mononuclear cells, we found that the individuals with the GIMATS high score, the anti-TNF refractory patients, tended to have lower amounts of monocytes in the blood, indicating that there's more recruitment of these blood monocytes from blood to tissue. One of the limitations, I think these findings have kind of held up in subsequent similar data sets that have been generated, but this was early days in single cell, and we were performing early single cell isolations. And so what we're moving toward at our lab, and we haven't published on this yet, are moving toward fixed samples, often from paraffin tissues, that we speculate, first of all, it's cheaper and it's easier. But we speculate may provide a more accurate estimate of relative cell abundances. It's just not going here. Okay. So the nod to risk lows, as I said earlier, are loss of function with respect to sensing this mirror multipeptide. And so it's a little bit counterintuitive at first. That was not the result we wanted 20 years ago when we first discovered this. And so it's complex, and I think we want to kind of make a story about these innate immune cells. This is from a paper that was published by Ruslan Medjitov at Yale a few years ago, where he really was focusing on this myeloid stromal crosstalk and the growth factors from the fibroblast, CSF1 or MCSF, will then activate the myeloid cells, resulting in growth in the myeloid cells, and in turn, the myeloid cells in homeostatic, healthy conditions will then shut off CSF1 expression. And conversely, the other thing happens with PDGF in the other direction. And so these myeloid cells or macrophages are enormously diverse, incredibly important in Crohn's disease. And one of the phases of understanding macrophage function is that it has many different functions in response to injury, infection, pathogen responses. So understanding the timing of these macrophage functions we think are important. And Ashika Nayar, who was a graduate student in the lab, wanted to look at this, since we showed in the GIMATS paper that there is recruitment of blood. I mean, it's well known that the ileal, the tissue ileal macrophages are even in health, but especially in disease, are constantly recruited from these blood monocytes. And what Shika had shown was that if you culture CD14 positive monocytes for quite a while, what you will see is that this is based upon the single cell data. If you look at the top on this, that's peripheral blood mononuclear cells. The bottom is tissues. You can see that there's expression of NOD2 in not just the myeloid cells, but also the fibroblasts. And that she defined a signature of that last row of the activated fibroblasts where there's very robust expression of a pro-inflammatory myeloid stromal niche that contributes to disease. What she then did was took the single cell data and stratified by NOD2 genotype status. So about a third of Crohn's patients will be carrying these NOD2 mutations. And she then looked at differential gene expression between the NOD2 carriers and the NOD2 non-carriers in our single cell data, took those lists of genes and performed pathway analysis, saw a lot of NF-kappa B signals. And interestingly, one of the pathways that she really implicated was that there was differences in STAT3 activation in the NOD2 carriers. And so what we implicate at the end of this paper is that we think that it was mediated through IL-6ST, which again is upstream of JAK1, which is now one of the major new drugs from the treatment for Crohn's disease. And so just to close out the NOD2 genetics, the fact that you have these 2 to 4% minor allele frequencies in NOD2, is there positive selection for NOD2 in European ancestry individuals? And our IBD at-risk population, is that positive selection mediated through at-risk populations that are then protected from mycobacteria? So to close out the myeloid stromal crosstalk story in Crohn's disease, you have crosstalk either by macrophages simply interacting with fibroblasts and tissues or mediating transdifferentiation. There are two different sources of cytokines or growth factors that are important in the myeloid cells. The homeostatic one, MCSF, or a GM-CSF, which is of high interest in inflammatory bowel disease. Our work has implicated the key role for blood monocyte recruitment and differentiation in tissues, and that the genetics provides another source of perturbation of these two cell modules. And then in terms of further complexities, it's not just recruitment of monocytes, it could also be recruitment of hematopoietic stem cells. And finally, obviously, in terms of the role of adaptive immune cells, which are important, the interaction between these innate lymphoid cells and IL-23R, which is one of the major associations in IBD, I think are important. A kind of another point to be made is that there is a striking overlap between the GWAS common variant loci that we've identified in IBD. This is kind of an old summary of this, and mycobacterial disease. And so when you think about the genetic architecture of these complex diseases, they've evolved in the context of these historically significant pathogens. And what was striking when we did this particular GWAS, identified 163 loci, is that there was overlap between these red genes or monogenic forms of mycobacterial susceptibility. But even more striking than that overlap was the overlap between IBD loci and these GWAS that were being published from Asia on multigenic forms of leprosy. And kind of the punchline to this story, I'm having some issues here, it's not, is that published from the Asan IBD Center a year ago, was that if you have a higher polygenic risk for leprosy, you have a decreased risk for Crohn's disease, again, arguing for this genetic evolutionary selection shaping the modern diseases that we see today. So in terms of Crohn's disease versus ulcerative colitis, we talked about the differences in tobacco. There are loci that are shared between Crohn's disease and ulcerative colitis, of which the IL-23 pathway is significant. And Another IBD general locus common between CD and UC is the ROS, reactive oxygen species, which affects both myeloid cells and epithelial cells. There's been, over the last many years, there have been a lot of these studies looking at the microbiome. And in general, I think the microbiome plays a greater role in ulcerative colitis than it does in Crohn's disease. The MHC class 2 associations are much greater in ulcerative colitis than in Crohn's disease. And in contrast, the NOD2 associations that I talked about are bacterial general. So I don't think that the specific types of bacteria play as big a role. I think it shaped the genetic architecture, but it's not so big in terms of managing or driving disease currently. And then in terms of innate immunity, this is where Crohn's disease really is distinguished from ulcerative colitis. We've talked a lot about NOD2. We've talked about the relationship. You have granulomas in Crohn's disease, so it's more macrophage driven. But I would argue that ulcerative colitis is much more mast cell driven. So this is not our work. This was published in Immunity a few years ago, where they basically took common variant GWAS and they overlaid them with the GWAS loci with the chromatin accessible regions across from a mast cell line data set that they developed. And so they sorted the different traits on the x-axis based upon the overlap fraction between the GWAS loci and the chromatin accessible regions within a mast cell line. And Behçet's disease was on the top. Things like allergic rhinitis, again, not surprising. But importantly, ulcerative colitis was like the number 4th highest complex trait, which overlapped with these chromatin accessible regions within a mast cell line. So I think this provides strong evidence that mast cells play a driving role in ulcerative colitis. And so our contribution to this literature was really looking at mast cells and fibroblasts in ulcerative colitis. And so the story is focused around a nominal genetic association that reported a couple of years ago for a gene in mast cells called MRGPRX2, which is a GPCR receptor protein. And it's one of the major means of activating mast cells via non-IgA mediated activation. And so we had seen a nominal association in this protein altering variant in MRGPRX2, and then Jinjing Dong at Johns Hopkins had published in Nature that the mouse ortholog of MRGPRX2, these individuals were protected against developing injection pseudoallergic reactions to these cationic drugs that were injected subcutaneously. And so the nature of this reaction kind of is similar to the reaction that we see to the inflammation that we see in ulcerative colitis, where there's a continuous superficial inflammation that always involves the rectum and can proceed proximally in a variable distribution. And so given that, we were, again, not sure if we wanted to move forward on this. And then this paper was published by Zhu et al. About 2019, where they performed cryo-EM with the rhodopsin receptor to define the structural features of the phosphorylation codes within those intracellular loops of GPCR proteins, and what would result in arrest and recruitment to then downregulate the GPCR signaling. And so the structure that they inferred from the cryo-EM is defined here. You have long and short arrest and recruitment codes that are going to then stop the signaling of the GPCRs. The P residues refer to a serine or threonine allele. GPCR-related kinases are serine threonine kinases. And you can see that if you have the association that we saw was for this in the first intracellular loop of MRGPRX2, where the ancestral allele is an asparagine at codon 62, and the minor allele, which is protective, is a serine allele. And so with the serine allele, you would be predicted to then result in a gain of an arrest and recruitment phosphorylation code. We then in this paper then show this in overexpression studies in 293 cells that to a wider range of agonists at MRGPRX2, we see increased recruitment and decreased signaling with the protective serine allele. In addition, if with that demarcation line that you see in ulcerative colitis, if you take biopsies, develop these laminar propionate mononuclear cells from either the inflamed regions in red or the uninflamed regions in green, and stimulate them with an MRGPRX2 agonist, you see a marked increased secretion of proteolytic enzymes, here carboxypeptidase measured, in the inflamed regions because they're gamma primed. So within the inflamed regions of ulcerative colitis, you see a marked increased capacity to secrete these proteolytic enzymes with MRGPRX2 activation. And so this results in a feed-forward loop with a risk allele disparaging 62, where, and then the final point is that what we think, again, we show through these expression studies, that the critical native cytokine activator for MRGPRX2 is PAMP12, which is a proteolytic product of adrenomedullin, which is produced from both the epithelial cells as well as activated fibroblasts. And so this invokes a feed-forward loop, which we think could be contributing. And then we're now looking at approaches to then block MRGPRX2. There are small molecules that have been developed by various biotech to potentially treat ulcerative colitis. Similar to the Jaimat's paper, the Martin et al. Jaimat's paper, this is an analogous study that was led by the pediatric groups, Ted Denson, Jeff Hyams. Greg Gibson led the analysis of this particular work, where you're basically taking at the initial diagnostic colonoscopy of children with ulcerative colitis and following them over the course of 1 year, performing a bulk RNA sequencing. And Greg's group developed a particular list of genes that in together resulted in a capacity to separate those individuals that had a colectomy by week 52 versus did not have a colectomy. And so our contribution to this was to then project that gene list that Greg's teams developed onto single cell data sets that we had from ulcerative colitis. And so the big contributors are, so this allows us to define kind of the genes that in a predictive manner, we're able to then predict a high risk for colectomy within a year. And so we can see in the yellow groups of genes is that innate lymphoid cell 1, we're showing evidence for association for the future risk of colectomy. These plasmacytoid dendritic cells that are interesting also predict early colectomy, which we thought was very interesting. And then finally, in terms of the epithelial cells, in this particular single cell analysis, we were able to divide the epithelial cells into secretory and non-secretory epithelial cells. And the secretory epithelial cells appear to correlate in a predictive manner a higher risk for colectomy within 1 year. And so the third two-cell module that I'll talk about are epithelial stem cells and lymphocytes. And epithelial cells are not well targeted in inflammatory bowel disease. And so a particular type of one of the kind of general class of genetic associations that we see in inflammatory bowel disease are this lack of these defects in NADPH oxidase to generate reactive oxygen species. Oftentimes, these people will present as kids with infections from chronic granulomatous disease, these mutations in either myeloid cells or epithelial cells that can result in IBD. And so, our contribution to this occurred earlier this year, where we basically took one of the NADPH oxidases that are expressed in epithelial stem cells. And the early part of this paper, we performed single cell RNA analysis of murine enteroids from the NOX1 knockout versus the wild type. We show that TNF, that the knockouts had absolutely zero capacity to secrete ROS secretion with TNF, and they also inhibit M cell signatures. One of our key findings in vivo in the knockout, which was very gratifying to us, is that if you take the NOX1 wild type versus knockout, the knockout to the right have these lymphoid aggregates between the base of those epithelial crypts and the underlying muscularis mucosa. And so that's one of the pathognomonic features of inflammatory bowel disease. And so we were able to recreate this in the NOX1 knockout. We quantitated this by immunofluorescence. And so what we think was happening here is that to the left, this is from a review article from Hans Cleaver's group. To the left, we're showing these LGR5 positive stem cells, those epithelial cells at the base of the crypts. To the right are the differentiated cells, and the middle column are kind of the intermediate types of cells. And so what we showed in our single cell data is that the NOX1 knockouts are characterized by these aberrant differentiation of the epithelial crypt cells. You result in these CCL20 expressing LGR5 cells, which then CCL20 then results in it's a chemokine that results in the recruitment of CCR6 expressing lymphocytes. So we're currently really expanding our single cell analysis on existing paraffin tissues using fixed tissues I alluded to earlier. So to conclude this part, modules, atlases, and emerging platforms, I've given you 3 examples of 2-cell modules. The myeloid stromal crosstalk and transdifferentiation, which is very unique to Crohn's disease. The mast cells, the non-IgE mediated means of activating mast cells through MRGPRX2, and the crosstalk between the agonists and the receptors, we think play a role in ulcerative colitis. And then we talked about aberrant differentiation of NOX1 cells and their capacity to recruit in vivo CCR6 lymphocytes. And so the next phase of what we're doing is we're now really expanding our single cell analysis on clinically attained fixed tissues, as well as proteins in larger patient numbers. And then we're just beginning to do spatial transcriptomics. We've decided to go with the Xenium platform at subcellular resolution. So finally, in terms of just a couple closing comments on genetic diversity, from a genetic perspective, one of the biggest priorities for the IBD Genetics Consortium moving forward is really to focus and recruit on diversity. So in the IBD field, we have well over 100,000 European ancestry individuals that have been recruited, and probably 2 to 3% of that in Africans. And yet, from a scientific perspective, this is incredibly valuable because recent African populations have the most genetic diversity, and they have the shortest linkage disequilibrium blocks, which is useful for prediction. And so understanding the differences in genetic architecture across populations, I think, is incredibly illuminating. This was work that was led by Carl Anderson's group at the Sanger Institute. And we're plotting on the y-axis the fraction of the variance explained for various genetic loci. And to the left are European ancestry variant estimates, and to the right are East Asian variants in Crohn's disease. And the blue alleles, NOD2 is monomorphic in Far East Asians. The brown alleles show where you have common variants, where you have markedly different effect sizes across populations, and the light yellow or tan alleles show similar effect sizes. So you can see that for many of the biggest effect alleles, we see markedly different effects across populations. And so just highlights, how much we can learn by going across different populations. And so another value of, besides just trying to understand the biology, is, let me just start by giving you the results. So the BioMe generated polygenic risk scores. So we did not specifically ascertain for IBD. This is just random ascertainment across a health system. And so basically our capacity to develop polygenic risk scores are improved if we use multiple cross-population data sets, as opposed to if we just simply focus on one population. And so this is the result from BioMe. We had a few hundred IBD patients. The green is Ashkenazi Jewish. So basically on the x-axis, we're plotting the polygenic risk score percentile. So we're going to higher scores of the polygenic risk score on the x-axis. On the y-axis, we're plotting the frequency of disease. So you're going to have a positive correlation with higher polygenic risk scores. But if within a population, so if you basically look at either the green curves or the blue curves, the bolded curve refers to where we basically use both European ancestry and African populations to develop the polygenic risk score. And you can see that the bold curves have improved performance of the polygenic risk score compared to the non-bolded curves, where we basically simply compare European to European. And then we took that same set of markers and then replicated in the UK biobank. And so It basically really highlights the value of these shorter linkage cystic component blocks, especially as we start integrating the genetic association from GWAS with epigenetic data on a single cell basis. And the final kind of point that I want to make is that this was a wonderful collaboration between Ron Doe here at Mount Sinai and the Penn team, Dan Rader and Scott Damrar. It shows the value of looking across populations. There's a particular amino acid variant, the V122I, within transthyretin, which has been associated with heart disease. And our contribution, and led by Ron, was really to look again in this randomly ascertained biobank cohort. The estimates that we got from BioMe were the same as had been previously reported in the literature, about a 1.6 to 1.8 odds ratio for heart failure. But importantly, most of these patients were undiagnosed. And so what Ron is now working on is, and you can treat this. So this is a case of congestive heart failure, where if you diagnose this early and prevent the accumulation of amyloid within the heart, you can potentially develop specific precision therapies for individuals who carry this particular mutation. And so Ron is now working on predictive models to then decide, how do we push? I mean, short of just sequencing everyone from the beginning, how do we push and target those individuals who might benefit for this more precision therapy for heart failure? So to conclude, for a wish list for biomedical research and precision medicine, really our goals are to diagnose early, to prevent delays in providing the most effective treatments. I'm not personally doing this, but there's obviously enormous growth in using digital continuous monitoring. One of our goals is that there's no digital divide across individuals of different resources. I think we should broadly adopt the cancer paradigm with respect to broad-scale research involvement. Early diagnosis is key, and early treatment and development of complete remission. The genetics in clinical care, we've done a little bit of this in inflammatory bowel disease with respect to these high penetrance IBD mutations. I don't think it's something that can be done on a trait by trait basis. It needs to be done across the health system. The genetics is obviously not determinative for most diseases. We have to compare it with multiple other omics, and we're getting very interested in really mining the existing tissues that exist in populations. And number 6 is really a challenge, is that if the goal is to develop better therapies, the concept of clinical trials continues to be incredibly challenging. How to do this, how to accelerate this to get better treatments for our patients is really a major goal moving forward, but it's a major obstacle. So in conclusion, I'd like to thank my lab. I'd like to thank my funders. The NIDEK has been incredibly generous, the Helmsley Trust, Regeneron. And then it's been my privilege to work with the Genetics Consortium for many years. The single cell has been done in collaboration, both in my lab as well as Mary Murad's lab. Continue to work with the BioMe group, Yuval Itan and Ron Doe. We've worked internationally very successfully, led by folks like Carl Anderson. And then we have a great IBD group at Sinai. So again, thank you for your attention. I'd be happy to take any questions.
Thank you, Judy. That was fantastic. If you want to take your slides off of slideshow, I will clap and thank on everyone who is online. And I will ask a question first from the chat. This came up actually halfway through, and I was going to interrupt you, and I thought I shouldn't. And I will just read it out. You mentioned that high GMAT module score is associated with non-response to anti-TNF therapy. Would peripheral monocytosis correlate with high GMAT score?
Yes, it does, but it's negative correlation. So basically you have recruitment. So the stromal cells in the tissue are producing large numbers of chemokines, which then recruit monocytes. So it's a negative correlation. So it's not monocytosis. It's actually draining of the peripheral blood monocytes into the tissues.
Yeah. And so before I ask the next one in the chat, maybe I can just ask you one, because on your wish list slide, it was quite interesting because it relates to much what we think about for clinical implementation. And there was two things I was thinking of asking you that were on there: the broad testing, or the broad scope of testing, and then the omics. I guess I was intrigued by this study you showed that a PRS score for leprosy is associated with decreased risk for Crohn's. And I guess that would argue that you can't, the future is unlikely to be panel testing for each of the PRS scores for each of the diseases. It's going to be, I presume you're an advocate for whole genome sequencing and then figuring out from there each of the risk scores. I presume that's what you would think, yeah?
Yeah. I mean, there's an old story that Eric Lander told me where it was at the beginning of the Human Genome Project, and like they would have all these groups. One was doing chromosome 1, another was doing chromosome 6. And like the idea is that we're all just seeing different parts of the elephant, and that we really, we need to scale this, and it just makes sense. You can't do it, subspecialty clinic by subspecialty clinic. It really needs to, in my opinion, it really needs to be within the health system. Kind of what Mount Sinai is doing. So Alex Charney and Girish Nadkarni here at Mount Sinai are leading this charge on the Mount Sinai Million, where the DNA is being isolated in CLIA space. And so we're going to do research grade sequencing, which is very different than clinical grade sequencing. And that if you estimate that 5 to 10% of folks are going to have actionable results, that seems the way to go if we're going to go in this direction.
And I guess you would also advocate for both DNA and RNA sequencing if it can be done?
Well, I mean, the GI tract, we have the luxury that for inflammatory bowel disease, I think that bulk RNA sequencing at the time of diagnosis, I don't know how I feel about that, whether that is something that should be done at the time of diagnosis, if we could somehow prove its efficacy and then get funding for it. I think the capacity to crosswalk across different data types, we actually may get there, in time. So that's, I think, particular to inflammatory bowel disease. In general, I've been a little bit disappointed with how much we've learned from peripheral blood single cell analysis, as opposed to tissue. That's much more tractable than tissue-based sequencing, except in cancers. Obviously, cancers are different. And again, that's the model that I think we can all point to, is kind of the cancer paradigm at the time of diagnosis. Do a complete workup, really personalize that. I think it's worked most successfully in cancer. So DNA, yes. RNA, I think probably we're not there yet.
Yeah. And so another question from the chat. So you mentioned IL-6 STAT3 pathway activation in Crohn's disease. Did you inhibit the pathway with IL-6 JAK or STAT3 inhibitors? And if so, did you see a response with respect to reduced inflammation?
So in the Nayar et al. Paper, what we did was we blocked IL-6 ST. I just went over that quite quickly. So the IL-6 ST, so anti-TNF is going to block the NF-kappa B pathways. And so in the non-responders, what are the pathways that are continuing to be active? And what we're trying to show in Nayar et al. Is that I think it has to do with IL-6. And unfortunately, IL-6 has not, blocking that pathway has not worked. There's been problems with intestinal perforations. And so it's IL-6 is just this general growth factor. And so we blocked IL-6 ST in that paper, which is, we think, more specific. We tried to engage various pharma companies in this. We weren't able to kind of get that off the ground. I think part of it is that IL-6 ST is upstream of JAK1, and JAK1 is just a blockbuster drug, new drug for inflammatory bowel disease now.
Yeah. And maybe a technical question. You mentioned this, that you were doing a lot of single cell on fixed specimens on FFPE, and you said you haven't reported on this yet, but can I ask, is that using the 10x, the fixed chemistry from 10x, or are you doing that with your own? And you're having success with that, yeah?
Yes. It just makes sense. I mean, you're just basically, so our best data, we've gotten some ileal, our best data is from liver. Liver is a very clean tissue relative to the gut, so I'm still not 100% sure it's going to work completely well in gut. But liver, we've done quite a bit, very clean data. It kind of makes sense that you're going to get more accurate estimates of cell abundance compared to kind of the old cell dissociation approaches. You lose kind of splicing data. We had been using RNA velocity splicing to predict RNA velocity and cell fate a little bit, but I'm not sure how rigorous folks think that is. So
yeah, it's funny. We've switched to the fixed and have the same issue, like lacking the splicing, lacks velocity, and I guess you change your. I mean, there'll be, I think, comparator algorithms, and we've generated both data sets now, nuclear sequencing and cell sequencing, to try and figure out the difference between them. So that was interesting to hear. Yeah, that was a fantastic talk. If there are no more questions in the chat, Judy, I'd like to thank you. I think it was very relevant what you said to many of the things ongoing here: the biobanking, it's the tour de force talk on the genetics and genetic risk and PRS, and ultimately, our goal, I'm sure, the same as your goal, is to turn these things into a clinical reality and try and make that happen.
Yes, for sure. Thank you so much for the invitation.
Yes, thank you very much for speaking. I appreciate it.
Sure. Bye.
Bye.